So we have discussed about immunology. Immunology is the science or the subject in which we study about immune responses our immune system have, and the immunity is a term for the system or the power our body have to protect us from the foreigner or foreigners or the microorganisms which can cause disease in our body. So there are various kinds of cells, tissues, organs. For example, cells, for example, we have macrophages, we have neutrophils, right? We have mast cells, T helper cells, T cytotoxic cells, B cells, and all these cells are responsible for protecting us somehow. Okay, for example, macrophages and neutrophils are phagocytes. So they can engulf, they can digest the microorganism. Second type of cell, for example, B cells. B cells will mature into plasma cells, and these plasma cell will secrete large number of antibodies. These antibody will bind to the pathogen or the microorganism and will neutralize that particular antigen or pathogen by binding to them. And also these antibodies will help in phagocytosis so that macrophages can recognize these antibodies because they are having specific receptor for the binding to these antibodies. Once they will bind to the antibodies which are already bound to the pathogen, so that antibody antigen complexes can be phagocytosized. This process is known as opsonization. We will discuss this also. Okay, so antibodies can work as opsins, and the process of phagocytosis with the help of antibodies is known as opsonization or uptake of that particular pathogen. I will tell you in detail. We will face this particular term in upcoming text. Okay, there are some tissues and organs. So we will discuss these tissues and organ one by one. There are primary lymphoid organs, secondary lymphoid organs, for example, bone marrow, okay, spleen, thymus. All these are important. They are present in our body and they are responsible in mounting an immune response so that we can be protected from foreign pathogens or microorganisms. And all these cells, tissues, organs, they carry out this activity and they will together constitute an immune system. So all the cells, tissues, and organs, if they are combined, they are known as immune system or the system of our body or the power of our body which will protect us from diseases. Immunity is the typical, it will be divided into two categories. One is known as innate immunity and second is known as adaptive immunity. Now we will go into the details of these two. A kind of immunity. So I told you that immunity is the power our bodies have to protect us from various diseases. And you know, immunity can vary from person to person. Immunity depend upon many factors. For example, it depend upon age, it depend upon the nutrition status, it depend upon the food we consume every day, it also depend upon the environment in which we live. So immunity is a power and it can be categorized into two main classes. One is known as innate immunity, second is known as adaptive immunity. So innate immunity is that power which we have since birth. Okay, so when a newborn or a baby is born, so it is having a power or a immunity that is known as innate immunity. And when it will survive in the environment, it will develop or it will experience some pathogens, some microorganisms, some infections, and these infections will be kind of experienced to that particular newborn or newborn baby, which will help them to develop the second branch or second category of immunity or the power. This is known as adaptive immunity. So therefore, the innate immunity is the same in all of us, but we are we are different in terms of adaptive immunity because we have different experiences of our infections we have faced in our life, right? So adaptive immunity will define an organism. Therefore, it will depend upon the food, the nutrition status. If we take more proteins, so obviously our immune system will be strong because we know antibodies are mostly glycoproteins. So they can be if we are consuming a protein rich diet, it will help in the build of our immune system, that is known as adaptive immunity. Okay. Also, the second factor is age. Age is also very, very important. With age, we face many pathogens, various infections, and therefore our immune system will become stronger with our age. So one factor which is very, very important in this category, in this adaptive immunity, is called as memory response. So now what is memory response? Or simply you can call it memory. Okay, so memory of immune system is very, very important in terms of adaptive immunity. You need to note here that innate immune system, which is almost same in all humans or in all organisms, but we differ in terms of adaptive immunity because of this memory response. Suppose we have faced some infection or some microorganism sometimes. So what will happen? Our immune system will be activated in response to that particular pathogen. And some of the B and T cells will be stored when this infection will be resolved. Okay. So these stored B and T cell will act as memory cells, and they will help our immune system or our body when the same microorganism or pathogen will infect again. Okay, तो अगर किसी को एक बार इन्फेक्शन हो गया है तो हमारी बॉडी में कुछ बी और टी सेल स्टोर हो जाते हैं जो उस अगर वो दोबारा से सबसीक्वेंट इन्फेक्शन होता है सेम माइक्रोऑर्गेनिज्म का, सेम पैथोजन का, so our body will quickly recognize that particular pathogen and try to eliminate that pathogen very quickly. So this is known as memory response. So memory response is the one of the properties of adaptive immune system or adaptive immunity. It is absent in innate immune system, right? Now, before going into the details of innate and adaptive immunity, you need to understand there are two systems in our body. One is known as blood. In blood, we have different kind of cells. For example, RBCs, white blood cells, WBCs, and platelets we have studied in our 11th, 12th class, right? But we also have one more system which is known as lymph. So this lymph, just like blood, have a group of cells which will help us. So see this particular diagram, you can see two kinds of systems or the circulation here. One is known as blood circulation system. So we all have blood circulation system in all of our body so that blood can be supplied to any of the organ or tissue. Okay, but we also do have this lymph system. So lymph nodes, for example, these nodes are kind of, for example, you can see here, they are present all over the body, but they are patches. They are called lymph nodes. They are structures which are filled with lymph. The blood is not present in these vessels, and these vessels are called as lymphatic vessels, right? So we have two kinds of circulation in our body. One is called blood circulation, one is called lymph circulation, right? So we have blood vessels, we have lymphatic vessels also in which the lymph will circulate. Now, see this. This is quite interesting. You can see here. So if you look this particular, how this blood and lymph are exchanged. Okay, so we have vessels which are called lymphatic capillaries or lymphatic vessels, in which the fluid is filled is known as lymph. Whereas we also do have blood capillaries or blood vessels in which the blood will circulate. Now, because their endings are very, very close, so that they can exchange some nutrients in between them. Okay. So a few lines are written here. Human lymphoid system. Human lymphoid system is the primary organs. Bone marrow and thymus are shown here. So this is the bone marrow. Actually, our long bones are having cavities which are known as bone marrow, in which the uh, B cells and T cells are actually, the bone marrow is the site or the primary lymphoid organ in which the B cells are synthesized and matured. But the T cells, which are synthesized in bone marrow, supply to the thymus, and then they will mature in the thymus, therefore known as T cells. Right. There are other structures also. For example, Pierce patches, there is spleen, and there are lymph nodes we already have discussed. One structure called as thymus, where the maturation of T cells taking place is actually situated in the chest region, in the thoracic region. This particular organ is present just above heart. Okay, so the thymus, spleen, bone marrow, Pierce patch, all these are making another system of our body in which the lymph will circulate, and the cells, for example, B cells and T cells, which will help in protecting us from various infections and diseases. Now, if you understand this particular, so actually for reference, we are using this book, 7th edition I'm having, right? So we will try to cover maximum from this book, because this will remain our reference book. And so first of all, what is immune system? Immune system is evolved to protect us, for example, multicellular organisms from pathogens, because we know most of the pathogens, for example, bacteria and viruses are not living, they are connecting links between living and non-living, whereas bacteria, they are single cells, they are, okay. So multicellular organisms, including humans, we are protected from the pathogens with the help of our immune system. So immune system is a kind of power we have which will protect us from bacteria, viruses, and all other microorganisms which can cause diseases in humans or in other multicellular organisms. So first of all, we will discuss type of pathogens which can cause disease in humans. Okay, so we will move to, and their size can vary from viruses which are very small in size in nanometer. We know poliovirus, which is around 30 nanometer in size, is the smallest, one of the smallest known pathogen, poliovirus, and it can large as a giant worm, which is known as Dictyofilm renale. Okay, so this is a kind of parasitic worm which will perform infection in kidneys, which can have size ranges 100 centimeter in length and 10 millimeter in diameter or width. Okay, so one diagram is given in the first chapter itself. If you look into the, so this green colored cell, which you can see here, I'm not talking about the whole this thing. This is only this one. This is macrophage, right? So this macrophage is a kind of phagocytic cell which will engulf some bacteria. These bacteria are Mycobacterium tuberculosis. Okay. So it is also written here, a phagocytic cell macrophage engulfing a bacteria that can cause tuberculosis, are shown in orange. So it will engulf many bacteria, including Mycobacterium tuberculosis, but tuberculosis is having protective strategy. It can protect itself inside macrophages. So once the macrophage will engulf this bacteria, mostly what should happen, it should degrade or it should digest these bacteria. But if this is Mycobacterium tuberculosis, so this Mycobacterium tuberculosis is having one protective strategy. It can be engulfed by the macrophages, but it will protect itself. It will not be digested by the macrophages, and it will survive inside the cell as intracellular parasite, right? So that's how they are able to survive inside the macrophages and can cause the Mycobacterium tuberculosis or TB disease. So first of all, we will discuss few pathogens which can cause diseases in humans. So we can all the pathogens into the categories: viruses, fungi, parasites, and bacteria. Viruses are the smallest, followed by bacteria. Fungi and parasites can have large sizes. Okay, so for example, we have discussed in microbiology class also, poliovirus is responsible for polio disease, variola virus, for example, smallpox, HIV for AIDS, rubella virus for measles. Okay, there are a few fungal pathogens. For example, Candida albicans can cause this candidiasis. Tinea corporis can cause ringworms, and so on. Okay, then there's few parasites written here: Plasmodium, Leishmania major, and Entamoeba histolytica. They can cause, for example, Plasmodium is a causative agent of malaria, and there are bacteria which can cause disease. For example, we had discussed Mycobacterium tuberculosis, causative agent of tuberculosis. Bordetella pertussis is a causative agent of whooping cough or the pertussis. Vibrio cholera and Borrelia burgdorferi causes Lyme disease. So a few is written here, but there are many microorganisms which can cause disease. Not only microorganisms, few parasites can also cause disease, which are having large dimensions. Now, more diagrams are shown here, which are depicting the role of macrophages. For example, okay, so it is the same diagram we have discussed. This is a kind of parasite which will cause disease or infection. Okay, then as I told you, so there are group of cells, tissues, and organs which can cause disease, which can protect us. This group of cells, tissues, and organs can protect us from many diseases, right? So the cells, all the cells which protect us from the invaders or foreign pathogens, they are derived from hematopoietic stem cell. First of all, these hematopoietic stem cell can be differentiated into two groups, two categories. One is known as myeloid progenitors and second is lymphoid progenitors. Okay, as we have discussed the two systems, one is known as blood circulation system, second is known as lymph circulation systems, lymphatic vessels in lymphatic. Okay, so this lymphoid progenitor cell, these cells will constitute the group of cells which are present in lymph, whereas the myeloid progenitor cell will lead to the development of those cells which are present in our blood system, right? So, and one thing is common, myeloid progenitor cell as well as this lymphoid progenitor cell, they can give rise to dendritic cells. So here also they can give rise to dendritic cells. Here also they can give rise to dendritic cells. Now what are dendritic cells? Dendritic cells are a kind of antigen presenting cell. We will discuss this in detail because dendritic cells are known as best antigen presenting cells. There are few other antigen presenting cells in our body. For example, macrophages can also work as antigen presenting cells. B cells can also work as antigen presenting cells, but they are having few major characteristics which will make them the best antigen presenting cells. These dendritic cells are okay, but they can be derived from both the lineages, either myeloid progenitor cells or from the lymphoid progenitor cells. Second thing is, in the lymph, there are natural killer cells, T helper cells, T cytotoxic cells, and B cells, which will give rise to the plasma cells. And these plasma cells will secrete large number of antibodies. Also, this myeloid progenitor, which is actually giving rise to the blood cells, in which you can say there are monocytes. These monocytes, if they move into the tissues, they're coming out of the circulation, blood circulation, then they will give rise to macrophages. The monocyte, macrophages, neutrophils, eosinophils, mast cells, basophils, and platelets. So platelets are actually not complete cells. These are cell fragments because they are derived from megakaryocytes, which are big cells. And if they are cleaved into small fragments, and then they are known as platelets. And we all know red blood cells, erythrocytes. So all these cells are present in blood. All these cells which are listed here, T helper cell, TC cells, B cells, and natural killer cells, they make the main WBCs. So they constitute 20 to 40% of WBCs, okay? And they are responsible for immune responses. In fact, neutrophils and macrophages are also involved in immune responses, but they are part of innate immune system, whereas all these cells will be covered in adaptive immune responses. Now, so we have already discussed about pathogens, for example, viruses, protozoa, there's fungus, some toxins which are secreted by these microorganisms. Some of the cells can be developed into the cancerous cells. These are called as transformed cells. There are parasitic worms, heterogenic procedures. So a human is prone to all these infections, right? So all these agents are kind of threat to the individual, so that we can have infection as a result of any one of them or all of them. So body is continuously exposed to many infections by these microorganisms or these agents. Cancer cells, toxic molecules which are secreted by some bacterial strains or some microorganisms, which can also cause disease-like symptoms, for example, toxic shock syndrome, fever, and all these things, illness. Even therapeutic drugs can also lead to kind of threat to the human, and they will come under this particular class, iatrogenic procedures. They are comprising therapeutic drugs and the procedures which are involved in the treatment of patients, right? Now, our immune system, whether it is innate immune system or adaptive immune system, so before going into the details of these two systems, we need to know few characteristics of our immune system. So our immune system have the power to recognize self versus non-self. What it mean? It mean there is a need for self-recognition. Our immune system should know what is belonging to our body. So it should not mount an immune response which is a part of our body. A wide variety of organisms, that is microorganisms or other organisms, for example, parasites, that associated and their associated molecules can pose a constant threat to the human body. They can cause infection in us. The human immune system is a kind of defensive mechanism that identify and neutralize these threats. Therefore, it is able to distinguish non-self organisms and their molecules from the self molecules. So our immune system have this power so that it will not mount an immune response against the self molecules, but it will mount an immune response if it is non-self or it is foreign, if it is coming from outside. To remove these infectious microorganisms or toxic agents, right, which are non-self. Now, our immune system actually consists of 3 layers of defenses, which will protect us from all of these: fungi, toxin, heterogenic procedures, bacteria, protozoa, viruses, cancer, and parasitic infections. So what are these 3 layers of immune system which are responsible for protecting us? Suppose they are microorganisms. So actually, if this is human, they are having 3 layers of protection. So one is known as barriers, right? Barriers, for example, our skin or mucous membrane is kind of barrier which will not allow these microorganisms to cause infection or to enter into the body. So these barriers are mainly skin and the mucous membranes, right? We will discuss this in detail. Second thing is innate immune system and then adaptive immune system. Suppose innate immune system try to eliminate this. So first of all, these barriers like skin and mucous membrane try to inhibit the entry of these microorganisms into the body. But if somehow they enter into the body, our innate immune system, which is the primary line of defense, they will try to neutralize these microorganisms. Okay, if they are not able to neutralize or remove these microorganisms and they will cause some kind of infection, then after some time these innate immune system cells, for example macrophages, they will secrete some cytokines, then it will activate the adaptive immune system. Okay, so finally the adaptive immune system and innate immune system work in coordination to remove the microorganisms out of body, to eliminate these infectious agents. So now we come to the innate immunity. Okay, so as I told you, there are 2 categories. One is known as innate immune system or innate immunity, and second is adaptive immunity. Innate immunity is the immunity or the power which is present since birth. So when baby is born, it is having some kind of immunity, and this immunity is known as innate immunity, right? It is consist of many factors that are relatively non-specific. Actually, innate immune system is not very specific system. It will mount an same response for all kind of infectious particles, but the condition should be all if they all are non-self. If they are self, the innate immunity will not work. Okay, consists of many factors that are relatively non-specific, that is operate against almost any foreign molecules. So they can work against any foreign molecule or pathogen or microorganism which can cause a disease. It provides the first line of defense. So innate immunity is considered as first line of defense, right, against pathogens. So they will try to remove the pathogen as early as possible. It is not specific to any one pathogen, but rather act against all foreign molecules and pathogens. Because as I told you, it can remove and it can operate against any foreign molecule or pathogen. And the response is quite similar. It will not differentiate uh, between different microorganisms or pathogens. It will try to eliminate any foreign molecule or pathogen once it will encounter. So it also does not rely on previous exposure to the pathogen, and response is functional since our birth. And this line is very important. It is having no memory. So as I told you, the only major difference between the innate and adaptive immunity, though there are other kind of differences also, as we will discuss, but one major difference is there is no memory response in innate immunity. So जैसे ही दोबारा उससे माइक्रोऑर्गेनिज्म भी अगर इन्फेक्शन कॉज करेगा, तो innate immune system उसी स्पीड से उसके अगेंस्ट immune response को माउंट करेगा. Okay, so it will try to eliminate the same pathogen with the same frequency and same speed. Okay, so let us discuss this in detail. So first of all, we will discuss few differences between adaptive immune response, and then we will discuss so there is a table over page number 17 which is showing the differences between innate and adaptive immunity. They will help you to understand the differences. So first of all, if you talk in terms of response time, innate immune system will be activated after minutes to hours. So once an infectious particle or pathogen enter into our body, innate immune system will be activated within minutes, right? Within no time. Second thing is adaptive immune response. Adaptive immune response is a kind of third layer, right? As I told you, it is a kind of third layer. If you are not able to understand, see it again. Actually, we are having three layers of protection from and responses from the microbial invasion. So first of all, we do have skin and mucous membranes, which are kind of barriers between our body and microorganisms. Second is we have innate immune system, which is a primary immune response. And there is adaptive immune system, which will be activated after this innate immune system. So actually the innate immune system will be activated within minutes, but adaptive immune system will take days for activation. So it is quite slow. Second difference is the specificity. As we have discussed in the Pathfinder, it is non-specific, or there is only limited specificity for any pathogen. Actually, it will behave similarly for any pathogen which is non-self, which is coming from outside. But the adaptive immune system is very, very specific. It is highly diverse. It adapt to improve during the course of immune response. So जैसे immune response mount होता है, so it will try to cooperate, it will try to improve with the course of immune response also. The more specific antibodies will be produced in this immune response, in mounting of this immune response, right? This is very, very specific in terms of any pathogen. Third is response to repeated infection. As I told you, innate immune system is having no memory response. So it will be same each time. Each time a microorganism will come, it will behave similarly each time because there is no memory cells, there is no memory response. But adaptive immune system will act more rapid and efficient, effective way in second or third exposure. Okay, this is called subsequent exposure. When the same microorganism or same pathogen will infect the same host repeatedly, our adaptive immune system will be activated within less time and the response will be more rapid as well as it will be more effective. It will try to eliminate that particular pathogen as early as possible. Okay, so let's understand this, how this more rapid response is due to this memory feature, memory response. So there is a diagram also which will help us to understand this adaptive immune system. So see this, there's differences in primary and secondary response to an injected antigen reflect the phenomenon of immunologic memory. So this memory response is shown in this particular diagram. So what is this? This is the magnitude of immune response. So how many cells and antibodies will be produced in response to this particular pathogen, for example. This is the time. So time should uh, keep into mind, and there are two diagrams in the single graph. So this is half is the primary response, then there is secondary response. What do you mean by primary response? Primary response is the first exposure to any microorganism. सपोज कोई माइक्रोऑर्गेनिज्म है जिसने पहली बार हमारी बॉडी को इन्फेक्ट करा है, then this is known as primary response. Now see the differences. और दूसरी बात सेकेंडरी रेस्पॉन्स, जब वही सेम माइक्रोऑर्गेनिज्म दोबारा से हमारी सेम होस्ट की बॉडी को इन्फेक्ट करेगा, देन इट इज नोन एज सेकेंडरी रेस्पॉन्स, दोबारा से इन्फेक्शन. So now see the differences in these graphs. Now if you look this particular graph, let me annotate this so that you can understand this better. Now see this one. This is innate immune system, right? Innate immunity. So you can see within this 0 day, the day of infection itself, it will be activated within minutes to hours. And as it will, showing the peak, this innate immune system, it will coordinated by the adaptive immune response. Then this adaptive immune response will switch and this innate immunity will go down, and then finally it will resolve. And the main role is of this adaptive immune response in removing that particular microorganism. Okay. Now you see this. This is day 14 here. And you can say around here it is day 10 or 11. So actually, if you see this, so this graph is growing from, it is coming from here. So it is actually the adaptive immune response or adaptive immunity. It will start working after a few days from here. So you can say this is around day 5 or 6, and then day 6, 7, 8, 9, 10, and about day 14, it is at peak. So after 14, 15 days, our immune system that is known as adaptive immunity is showing the peak level, right? Whereas our innate immune system is at peak around or before our adaptive immune system start working. So around day 3rd or 4th, our immune system is working very fine, and then our adaptive immune system will showing the peak. So this peak is nothing but the number of cells which are activated in response to the infection or the injected antigen. Now see this, the secondary response. Secondary response is if we inject the same antigen or same pathogen again to the same host which were infected previously. So as we know, the innate immune system will not show much difference. It is very, very similar to the first one. If you see these two graphs, they are similar. But the difference is quite interesting if you note this point. So suppose the innate immune system is activated almost at the same time, because this is day 0 and this is the repeat antigen A. Previously, we have injected the antigen A also. When we repeatedly injecting the antigen A, you can see here that at day 0 itself, or after this a few hours later, because there were mammary cells which will help this adaptive immune system or adaptive immunity to work and see the mounting response, the differences. Here it is, this is the peak, but in the second case or during the secondary exposure, this peak is very, very efficient or effective. So this is known as effective immune response when the same antigen is exposed again and again. So innate immune system will not show much differences in repeated exposure of pathogen, but adaptive immune system will show the differences when it is the primary response and it is the secondary response. Now, what is responsible for this difference? So this difference is due to the memory response or the memory cells which are stored during the primary infection or the primary response, and they are activated as early as possible when the same antigen will be encountered again. And during the secondary response, this is early and more effective. So the peak is showing the effectiveness, and this peak, which is actually start emerging from here. In the second case, it is emerging from day 0 itself, or the same day when you inject the antigen, injected with antigen. So it is rapid response, and it is more effective in terms of immune response, which is mounted against the same antigen. It is showing the memory response, right? Third is major components. If we talk in terms of innate immune system, so the major component which are responsible are barriers, for example, skin and mucous membranes. There are few cells, for example, phagocytes. These phagocytes can be neutrophils, which are present in blood, or macrophages, which are derived from monocytes. So monocytes are present in blood circulation, but once the monocyte come into the tissues or at the site of infection, they will be converted into the big cells. These are known as macrophages, or macrophages are the main phagocytic cells. Pattern recognition molecules. So we will discuss these also. Pattern recognition molecules or pattern recognition receptors. We simply can write them pattern recognition receptor, PRRs. So one kind of pattern recognition receptor, we will discuss TLRs, toll-like receptors. So we need to cover these topics, pattern recognition receptors and toll-like receptors. What is the role of pattern recognition receptors? So let me tell you this one first after finishing this table. Okay, so adaptive immune system. So main type of cells in the innate immune system are phagocytes, and those cells are having pattern recognition receptor over them. Whereas the adaptive immune system is having B and T cells. So also they are having natural killer cells, NK cells. So three type of cells are present in adaptive immune response, B, T, and NK cells. So you can write them here, B, T, and natural killer cells, right? So they will constitute our adaptive immune response, mainly B and T cells, right? Antigen specific receptors, because they are having very specific antigen specific receptors and antibodies which are present over B cells. B cells will be converted into plasma cells, and therefore they will secrete large number of antibodies. So plasma cells क्या भी? Plasma cells, B cells से जब क्या होता है इन्फेक्शन होता है तो बी सेल्स are matured into plasma cells. These are large cells which are having very active transcription and translation. These translation event will responsible for making large number of antibodies, because we know antibodies are glycoproteins, right? Proteins, and proteins are derived as a result of translation. So they will secrete large number of antibodies, whereas in B cells, the antibodies are not secreted, they are attached to the cell surfaces. Yes, students, if you have some query, please ask me. So this is a break for 5 minutes, and then we will discuss. You may ask your queries right now. If you have a query, please ask me. Okay, so there are very deadly diseases. For example, the smallpox. Oh, sorry, this is smallpox. Sorry. So this is smallpox. Okay, so they are very deadly diseases. In fact, they are pathogens which can cause severe infections, and they will leave the host disfigured for life, or even in some cases, the death even may result. Okay, so few diseases are very deadly and they should be eliminated. Uh, this is because of this subject that large number of diseases, for example, smallpox, diphtheria, rubella, measles, mumps, whooping cough, pertussis, polio. Rubella is actually so this is there is difference between this measles and this measles. So this is German measles. Okay, tetanus and Haemophilus influenza doing common cold. Okay, so if you see this number percentage of reduction, so smallpox, diphtheria, measles, mumps, and pertussis, polio is eradicated about 100%. Even smallpox, diphtheria, they are all eliminated as a result of discoveries and vaccines which are developed. Okay, so this is one of the major application of immunology in development of the vaccines against these deadly diseases and few other diseases, including this tetanus, mumps, rubella. They are also reduced, and the number percentage is given in this particular table. You can refer this one. So we have discussed there are barriers, there are innate immune system, there are adaptive immune system. Barriers is actually a component of innate immune system. They are helping host so that these pathogens cannot enter into the host body. So we do have various kind of barriers. Let discuss them in more details. So first is elements of innate immunity, right? So innate immunity have physical barriers. Physical barriers are first line of defense against microorganisms. They include skin and mucous membranes. This skin is present all over body except a few openings. For example, our mouth, it is the epithelium is called mucous membranes. These mucous membrane secrete some waxy substances, and these will entrap the microorganisms. Okay, most organisms and foreign substances cannot penetrate intact skin. If there is skin is intact, there is no problem. We can be protected. Okay, but if there is some injury or some kind of breach in this barrier in the skin, then microorganism can enter into our body and they can enter into the tissues and organs. Then they can do this infection. So where it was given, wait a minute. So I will try to find one diagram. It was given somewhere. Wait a minute, actually one diagram I missed it. Okay, 172. Oops. We'll come to this one, but actually I need one more diagram. Okay. Right, so it is here actually, page number 167, right? So actually, this is the skin, and below the skin there is other layers. If there is a breach or there is a damage to the tissue, suppose we are having some kind of injury or we are uh, having this tissue damage, so what will happen along with this particular thing? Suppose this is a knife or blade or some thorn. Few microorganisms, few bacteria can enter into our body. Okay, so the primary barrier which is protecting us against these microorganisms, against these bacteria, is broken, and therefore they will enter inside the tissues. Okay, so the tissue damage and bacteria cause resident sentinel cells to release the chemoattractant and vasoactive factors that trigger a local increase in the blood flow and capillary permeability. So now what will happen? These bacteria will enter into the body. Then this tissue damage, the cells which are damaged here, they will release a kind of substance which are known as chemoattractants and vasoactive factors. These chemoattractants will attract few cells from the blood circulation, for example, neutrophils, dendritic cells, macrophages, monocytes. Okay, these will come at the site of infection, site of entry of bacteria. So there are two other responses. One is local increase in the blood flow in this particular area where you will have this kind of damage in the tissue. If you have seen that if there is an injury, अगर कहीं पे आपको चोट लग जाती है, इंजरी हो जाती है या टिश्यू डैमेज होता है, तो वहां पे स्वेलिंग हो जाती है, स्वेलिंग और redness because there is an increased blood supply to that particular area. And because of more blood in that particular area, this is local increase in the blood flow in this area, it will result in redness. Okay, so this local increase is triggered by the chemoattractant molecules and vasoactive substances which are released by these damaged cells, these damaged cells in this tissue which is facing the injury or breach. Now, the second thing is capillary permeability. The vasoactive substances are those small chemical agents or factors which are responsible for increasing the permeability of these membranes, these capillaries in which the blood is flowing. We know with blood there are many different type of cells which are moving in blood in these capillaries. So what will happen, these vasoactive substances or factor will increase the permeability of these blood vessels or capillaries so that these neutrophils and other cells which are required at the site of infection will come outside the blood vessels. So there are three steps. One is the binding or the attachment. This can release some of the antimicrobial protein directly, or the cells will come out of blood vessels or capillaries by the process of extravasation. So extravasation is, so the first thing is the increased permeability. How this permeability is increased? There are endothelial cells which cover the blood capillaries. So you can see there are few gaps between them. This gap is increased, so that increasing the permeability of these blood capillaries. Due to the increased permeability, the extravasation will happen. Extravasation is the process by which neutrophils or other cells will come out of capillary into the tissue space or the site of infection. So you need to note these terms, extravasation, and once the extravasation is complete, this cell will come into the tissue spaces, will move towards the site of infection or where the bacteria are present in more number, and this process is known as chemotaxis. So binding, extravasation, and chemotaxis. So there are three processes which will require to move these neutrophils and other cells, for example, monocytes, from the blood circulation to the site of infection in the blood, from the blood into the tissue spaces. So extravasation is followed by, for example, neutrophils and other phagocytes are moving out of the blood circulation by this process of extravasation in between two endothelial cells because of increased permeability of capillaries, right? Then neutrophils and other phagocytes, they will migrate to the site of inflammation or infection, is known as chemotaxis. This chemotaxis is a resultant of this chemoattractants which are released by these uh, cells, which are damaged tissue cells. These cells will release some chemoattractant. They will attract these cells which are coming out of blood circulation to enable them to move at the site of infection, at the site of inflammation. And because there is more blood supply to this area, there is redness, there is more number of cells in this area, so that there is swelling. Okay, so these are few symptoms whenever there is a tissue damage or injury. First step is phagocytes and antibacterial substances will destroy bacteria. They will try to eliminate them. They will engulf them. So monocyte, which will come out of the blood circulation, will be modified into the macrophages, which are big phagocytic cells, which can engulf the bacteria and then digest them into smaller fragments. Hope you are able to understand everything from this diagram. If not clear, please let me know or ask your queries. You need to know these terms because these will come frequently. Extravasation, chemotaxis, and these two terms are very prominent, very common. All right. So physical barriers, which are first line of defense against microorganism, it includes the skin and mucous membrane. Most organism and foreign substances cannot penetrate the intact skin, but can enter into the body if the skin is damaged. So if there is a damage in the skin or the tissue, they can breach this barrier and they can enter inside the tissues. Okay. Otherwise, it will provide primary line of defense. This is a kind of barrier between the host and the pathogen. Understand? तो केवल जब हमें कोई इंजरी होती है या किसी काइंड की तरह का इन्फेक्शन या फिर हमारी स्किन या म्यूकस मेम्ब्रेन्स में जो इन्फेक्शन डैमेज होता है उसकी वजह से ही माइक्रो ऑर्गेनिज्म टिश्यू स्पेसेस में पहुंच जाते हैं. ओके, so most organisms and foreign substances cannot penetrate the intact skin. We understand this, but can enter the body if the skin is damaged. Right? Secondly, the acidic pH of the sweat and sebaceous secretions. दूसरी चीज क्या, एक तो हमने डिस्कस करा कि skin or the barriers which are present in our body. Second thing is our secretions, for example, sweat. Okay, and the sebaceous secretions. There are some sweat glands. There are sebaceous glands which will secrete some kind of waxes, right? So these secretions are having acidic pH, and this acidic pH will not allow the growth of microorganisms. And the presence of various fatty acid and hydrolytic enzymes, for example, lysozyme, which is present in our tears, which is present in our saliva, is also responsible for removing the microorganisms, right? For example, bacteria. So lysozyme will degrade the cell wall of bacteria. So therefore, it will inhibit the growth of most of the microorganisms. One more thing I will tell you, known as so we will go to page number 146 in our QA. 146. Actually, the diagrams which are present, which are given in this book, are very interesting. They are colorful and you can understand them because they are very clear. Okay. So actually the phagocytosis, the process of phagocytosis is shown here. Suppose there is macrophage and bacteria. So as we have discussed in our last diagram that the macrophages will move from blood circulation to the site of infection and they will try to remove the bacteria. Now how they will remove the bacteria? The bacterium become attached to the membrane in the evaginations. Evagination are, they are outside. Evaginations, these are called pseudopodia, and they will surround the bacteria. Bacteria are ingested and they're forming phagosomes. The membrane is covering this structure is called phagosome. Phagosome will bind to the lysosome. Lysosome, you know, suicide bags, and they are having mostly acidic pH. Okay, acidic hydrolases enzyme which work best around pH 5. So they will fuse, the lysosome structure will fuse and they will release the acid hydrolases. You can see these small dots are acid hydrolases enzymes which will degrade the bacterial substances. Bacteria is killed and digested by the lysosomal enzymes, degrade them, it will digest them. And then finally the products or the smaller fragments of bacteria, they are released by the process of exocytosis, right? So there are two processes, endocytosis and exocytosis. And in this process, the bacteria is digested and cleaved into smaller pieces. This process is known as phagocytosis because the phagocyte or the macrophages are also the smaller smaller phagocytes are called as neutrophils are also responsible for this process. But the major phagocytes, because they are having large sizes and therefore macrophages are major phagocytes. I want to show you one more diagram actually, but here it was given. Okay, right. It is given here actually 145. So I'm reading few lines. You need to understand this. Not only lysozyme, our skin do secrete few peptides which are responsible for killing the microorganisms. Okay, तो एक टेस्ट करते हैं उसमें क्या करा है इन्होंने so you on a fresh culture plate when you put your fingers you will see that there is no E. coli growth, right? What it showing? So it is showing that our skin do release few antimicrobial peptides which will kill the E. coli bacteria or some other microorganisms. So this peptide is known as antimicrobial protein is known as psoriasin. Okay, psoriasin is present. It prevents the colonization of the skin by E. coli, and it is continuously secreted by our skin. Skin secrete psoriasin, an antimicrobial protein that kill E. coli. Okay, actually the fingertips are shown here, but it is present all over our body. So it will not allow the E. coli colonization or some other microorganisms which are present in the environment so that they can colonize in our skin, on our skin. So our skin continue to release this particular antimicrobial protein and it will kill the E. coli. But it is seen that it will not kill some pathogens, for example, S. aureus, and therefore the S. aureus can do acne disease. So these acne is a result of the infection from S. aureus. There are other proteins and peptides, which are given in this particular table. Proteins, for example, lysozyme. As I told you, it is present in our secretions, for example, tears, saliva. It is also present in our respiratory tract. What is the function of lysozyme? So it will degrade the cell wall of bacteria. So clearly the glycosidic bond in peptidoglycan cell wall in bacteria, and due to which the bacteria will lyse, bacterial cells will lyse as a result of endoosmosis. There are other proteins also, for example, lactoferrin, secretory leukocyte proteases inhibitors, S100 proteins, as we have discussed, 1-picoresin, which is also present in skin, mucus, and glandular secretions. There one more, calprotectin, which is also present in the skin secretions, and it bind and sequester divalent cation, for example, manganese and zinc. If it calprotectin bind to the manganese and this amine and zinc, it will not allow the growth of bacteria and fungus. There are other proteins like defensins, alpha and beta, cathelicidins, and surfactant proteins, for example, SP, surfactant protein A and surfactant protein D. They also block the surface components, promote the phagocytosis of pathogens. So there are different kinds of proteins and peptides which are helping in this, as we have studied. These are acidic, these are having acidic pH, and therefore they will not allow the growth of most of the microorganisms. They will inhibit the growth of them. Similarly, respiratory and GI tract lined by mucous membranes. We know this. And this mucous membrane, because there are some secretions, they will entrap the microorganism. Suppose there is some microorganism, the mucous membrane secretion will entrap these microorganisms. The respiratory tract is also covered by cilia. So, cilia are hair-like projections which are present over these epithelial cell membranes. Once the microorganism is entrapped into the mucus, these cilia will move. This is known as synchronous movement. They will move simultaneously, cilia in one direction. So they propel mucus entrapped microorganism out of these tracts. So suppose in our GI tract, gastrointestinal tract, or in our respiratory tract, if some microorganism or some pathogen is entrapped, it should be removed with the help of the synchronous movements, with the help of cilia, and then we have sneezing or cough so that it can be removed finally from the respiratory tract. Similarly, the conjunctiva is a specialized mucus secreting epithelial membrane. Now you need to understand this. This is present in our eye. Okay, conjunctiva is a specialized mucus secreting epithelial membrane that lines the interior surface of each eyelid. To understand this, you need to see one diagram. So, hope this is visible to all of you. Hey guys, is this visible? The structure of the eye. So here it is showing. Okay, fine. So we have these lacrimal glands. Okay, just above our eye structure, and they're having ducts through which the secretions from these lacrimal glands will come into our eye, or the tears, right? These tears or the secretions of these lacrimal glands are also having lysozyme. Lysozyme is the enzyme which is responsible for degradation of these bacteria. They will kill the microorganism. Okay, so And also to remove all these dust and other microorganisms, there are lacrimal sac-like structure which is present here, and it will remove the dust or impurities from the eyes. Okay, so also if you are not able to understand this from here, so this lacrimal sac is connected to our nostrils, and then it will be released through our nose. Okay, so that's how it will operate. Okay, so opsonization I already told you. So opsonization is the removal of bacteria with the help of antibodies. So if antibodies are bound to any antigen, and the phagocyte, for example, macrophage is engulfing this antigen antibody complex, then this is known as opsonization. So we move back to our discussion. Similarly, the conjunctiva, which is a specialized mucosecreting substance or the epithelial membrane that lines the interior surface of each eyelid, it is kept moist by continuous flushing action of the tears. Tears are known as lacrimal fluid because they are coming from a gland, as I showed you, lacrimal glands. Okay, the tears containing the enzyme lysozyme, not only lysozyme, they also do have lactoferrin and immunoglobulin A, and this is antibody A, and thus provide chemical as well as physical protection. So it is having physical protection because they do provide a kind of barrier. And the second thing is the chemical protection because they continue to release these chemicals called lysozyme, lactoferrin, and antibody A. This antibody A is present in secretions, for example, tears, and also in milk. Microorganisms do occasionally breach the epithelial barricade, as we have seen in one of the diagrams. It is then up to the innate and adaptive immune system to recognize and destroy them without harming the host. This is very, very urgent because in this complete immune response, host should not face any kind of damage. So the innate and adaptive immune system will finally remove the pathogen, but they will not harm the host in any by any mean. In case of innate immune response, several antimicrobial chemicals and phagocytic cells provide protection against pathogens. So in case of innate immune response, so we have seen these antimicrobial chemicals, for example, lysozyme, lactoferrin, or we have studied many others in the, remember from this table, lactoferrin, psoriasin, calprotectin, defensins, cathelicidins, surfactant proteins. All these will help in kind of chemical mediators which are part of innate immune system, right? So now it comes to the chemical mediators. A variety of chemicals mediate protection against microbes during the period before adaptive immune system develop. And molecules of the innate immune system include complement proteins. So this is quite interesting. We have a complete chapter about complement proteins, right? So complement proteins are given around page number 187. So if you move 187, so we have a complete chapter known as complement system, which is sixth chapter of the QP. Okay, so there are many different type of proteins which are given in page number 188 on the next page only. So the proteins involved in the complement system include C1q, mannose binding lectins, ficolins. uh, there are C1 R protein, S protein, C4b, C2a. So you can remember C1, C2, C3, C4, C5, C6, 7, 8, 9. So these are major complement proteins which will create a pore-like structure in the membrane. So suppose this is the bacterial membrane. So the C7, 8 and C9, actually C9 will make a channel through the pore through the plasma membrane, and therefore it will disrupt the osmotic balance between the cell inside and outside of the cell, right? And it will lyse that particular bacterial cell, result in lysis. So there are complement proteins. There are cytokines. Cytokines are, for example, interleukins. Cytokines are proteins or small peptides which are secreted by immune cells so that they can activate each other. Okay, as we have discussed that the innate immune system is the first or the primary line of defense against any infection, then followed by adaptive immune response. So adaptive immune response or the cells of adaptive immune system should be activated by innate immune cells by secreting few cytokines. These cytokines will activate other branches of immune system. There are pattern recognition receptors, PRRs, I told you. For example, toll-like receptors, which are given in the next page. There is acute phase proteins, cationic peptides, enzyme, for example, lysozyme, and all these which we have discussed in the over page number 146, I think. Right. So all these are chemical mediators. Sorry, 146. All these are the proteins or polypeptides or kind of mediators which will help in innate immune responses. Then there is complement proteins. As we know, as I showed you that there is C1, C2, C3, C4, 5, 6, 7, 8, 9, and there are other proteins which are responsible for making pores in the plasma membrane or the cell envelope of bacteria. So these complement proteins are soluble proteins or glycoproteins, means they are proteins with the carbohydrate portions. In fact, antibodies are also glycoproteins that are mainly synthesized by liver. So these complement proteins are synthesized by liver, and then they will come into the circulation or in the blood, circulate in the blood and extracellular fluid, which are present outside the cells. They were originally identified by their ability to amplify and complement the action of antibodies. So what is the main role? Why they are called complementary proteins? Because they will help complement in the action of antibodies. They will help antibodies, they will help other cells in the phagocytosis. Therefore, they are called complementary proteins. We will discuss them in detail. We will when we will come to this particular chapter, right? Hence, the name is given as complement proteins. There are about over 30 serum proteins which can be called as complement proteins. Activation of complement protein in response to certain microorganisms result in the controlled enzymatic cascade, which target the membrane of pathogenic organism and lead to their destruction. So what will happen? They will activate in a sequential way, C1, C2, not necessarily that the C1 will activate C2 and then C3, but it is a random, so 188. So this is a random sequence of events. Okay, for example, first step is these antibodies will bind to the antigens, which are suppose this is bacterial surface. On bacterial surface, the antibodies are attached, so they are helping antibodies, right? Therefore, they are called complement proteins. So the complement protein C1q, MBL, and aficolin, they will come and bind like this, okay, like a bunch, and then other proteins will come, convertase and activators, for example, C1R, S, C4B, C2A, enzymatic mediators, C3 convertase, C5 convertase, they will come and they will activate. Finally, a structure will result that is known as, this is complex, MAC complex, membrane attack complex. C5B, C6, C7, C8, and rest is C9. So this is a kind of channel or pore which is inserted into the cell envelope or the pathogen envelope. And finally, the content of bacterial cell will come outside through this pore so that the bacterial cell will lyse. So this is known as membrane attack complex, right? There are few components of these complementary proteins. Their component will be released and they will act like alpha anaphylotoxins. Anaphylotoxins means they will help in inflammatory responses. Okay, and then they may help in the phagocytosis. So this process is known as opsonization. Okay, so they also work as opsonins or opsonization events. Fine. Finally, they help in the destruction of microorganism or pathogens. Then cytokines. Cytokines are a generic term, or the term which is used for low molecular weight and so they are low molecular weight soluble proteins or glycoproteins released by one cell population, and they will act intercellular mediators. Intercellular mediator means they will activate other cells. They are secreted by one cell and they will activate some other cell. For example, let me tell you two examples here so that you can understand this better. We do have interleukins. So interleukin is kind of cytokine, which are small. So always we write interleukin as capital IL. And we have interleukin 2, for example, interleukin 4. They're written like this. So we have two category of interleukins. One is known as interleukin 2, one is known as interleukin 4. Interleukin 2, what is the role of interleukin 2? It will be secreted by the activated T cells and they will activate, okay, wait a minute. So let me confirm, I do not want to tell this wrongly. Okay, fine. So there is interleukin 2, which are secreted by T helper cell 1 population, and they will activate other T cells, right? So they are secreted by T cells, they will activate other T cells. So these are kind of intercellular molecules. Now we have interleukin 4, which is the second interleukin. It will activate B cells. So both cytokines, interleukin 2 and interleukin 4, they are secreted by the same T cells, but they will activate different type of cells. For example, interleukin 2 will activate the T cells, interleukin 4 will activate the B cells along with other interleukins. For example, interleukin 5 will come, interleukin 13 will come, and so on. So there are other interleukins or cytokines also involved. And finally, these interleukin 4 will activate B cells. And once the B cells are activated, they will secrete. They will make plasma cells, and they will secrete large number of antibodies known as humoral immune response. And this is cell-mediated immune response, CMI. So both are kind of adaptive immune response. Okay, so that's how these cytokines will help in activation of other cell populations. It includes monokines, lymphokines, interleukins, interferons, and others. Okay, so interferons are kind of mediators. Suppose if there is viral infection in one kind of cells, so interferons will be secreted to virally infected cells. They will alert non-infected cells that there is a virus in the system, and they should protect themselves against these viral pathogens. So interferons are kind of secretory molecules or cytokines which are secreted in response to viral infection. And they will create an alerted state in non-virally infected populations of cells. Cytokines are required for immunoregulation of both innate as well as adaptive immune response. Cytokines की जो requirement है वो immune responses के regulation के लिए होती है क्योंकि बहुत सारी cells को एक population of cell can secrete large number of cytokines, and these cytokines can activate different kinds of other cells, either in the innate immune system or as well as in the adaptive immune responses. Interferons are cytokine made by the cell in response to viral infection. Okay, so it is given here, which essentially induce a generalized antiviral state in surrounding cells which are not infected. So interferons will be secreted by a cell which is infected with virus. They will go to the nearby cell and they will tell them, oh, there is an infection or there has been viral infection, so that other cell can make some kind of protective molecules so that they can be protected from the viral infection. Now, chemokines are small positively charged secreted proteins that have a central role in guiding the migration of various types of white blood cells. So chemokines, for now, there is another kind of molecules known as chemokines. We have studied cytokines, right? Chemokines are, okay, so first of all, we have discussed one diagram in which I showed you that neutrophils are coming out of blood vessels and they are moving to the site of infection. This is known chemotaxis, right? So the chemotaxis is a result of chemokines. So there are chemoattractants or the small molecules which will attract the cells towards the site of infection or where there is inflammation. These chemokines are small, positively charged secreted proteins that have a central role in guiding the migration of various types of white blood cells. So these white blood cells, for example, neutrophils, monocytes, etc., will move from blood circulation to the site of inflammation or injury with the help of these chemical attractants known as chemokines. They bind to the surface of endothelial cells, white blood cells, and to negatively charge proteoglycans of extracellular matrix in the organs, and they finally come out of the blood vessels. By binding to the G protein linked receptors, GPCR, on the surface of specific blood vessels or blood cells that are called endothelial cells, right? Chemokine attract these cells from bloodstream into an organ where the injury is actually happening and guide them to the specific locations within the organ and then help stop migration. So overall, the migration is having the central role of one kind of positively charged secreted small proteins which are known as chemokines, right? Now there are some receptors which are present over the surface of these cells which are moving, which are phagocytic cells which are moving from blood circulation and they are going towards the site of injury or inflammation. Now, first of all, we will understand what is PAMP, what is PRR. So PRR are pattern recognition receptors which are present over phagocytic cells. And PAMPs are pathogen-associated molecular patterns which are present present in over microorganisms or pathogens. So now there is a specific binding between PAMP and pathogen recognition receptors. Look into the diagram first. So this particular diagram we have suppose this one, effects of innate immune response to an infection. So this is a pathogen. So this is a bacteria. Bacteria is having various kinds of surface molecules which are present over its surface, right? Which will be recognized by the phagocytic cells, for example, macrophages, or other cells, for example, neutrophils, which are having pattern recognition receptors which will bind to these PAMPs. So PAMP and PRR will bind together, right? So we also have one more better diagram somewhere. So let me find first. See this. So as I showed you these arrows or these sorry. So as I showed you these structures, these are known as PAMP, pathogen associated molecular patterns, right? So in more details, they are shown in this particular diagram. Now look this. Suppose this is a gram-negative E. coli, right? So we know gram-negative is having two plasma membranes. One is outer membrane, one is inner membrane. There is a small peptidoglycan cell wall present between these two layers. And they also do have lipopolysaccharides, which are endotoxins. So these lipopolysaccharides or other structures which are present over the surface are actually known as PAMPs, pathogen-associated molecular patterns. For example, in gram-positive cell, there are different kinds of patterns. For example, they have peptidoglycan cell wall. This is S. aureus, gram-positive bacteria. So they are showing the surface, right? At the surface, there is a membrane, plasma membrane, and there is a 20 to 80 nanometer thick peptidoglycan cell wall. And over peptidoglycan, either they have glycoproteins or glycolipids, which are directly linked to the plasma membrane lipids, right? So glycolipids and glycoproteins, they are kind of patterns which are present in gram-positive bacteria. Similarly, fungus and viruses do have kind of patterns or the surface receptors which are known as pathogen-associated molecular patterns, or in simple terms, we will call them PAMPs. So let me, so these are known as PAMPs, associated molecular patterns, which will comprise of glycolipids and glycoproteins, for example, in gram-positive bacteria. And gram-negative bacteria, these are, for example, lipopolysaccharides, LPS, in gram-negative bacteria. So they will constitute this pathogen-associated molecular patterns. Now these patterns, because they are present over pathogens, pathogen-associated molecular patterns, right? They are present over pathogens or disease-causing microorganisms. Therefore, they should be recognized by pattern recognition receptors. Pattern recognition receptor, PRRs, they should be present over macrophages or some other kind of cells which will be acting as our immune cells, right? Macrophages will recognize these PAMPs. So look into an so that you can understand this pattern. Suppose in this diagram, what is shown. So this is a pattern recognition receptor that is also known as toll-like receptor, TLR. So toll-like receptor is actually present in macrophages or some other immune cells, which are cells of innate immune system. They are having TIR domain, which is intracellular, and they are having an extracellular domain. This is known as exterior domain or leucine rich repeat, LRR. They are having multiple leucine amino acid in this particular repeat. So this is also shown in this particular Revan model, right? They help in recognition of patterns. So what kind of pattern they recognize? Now see this. These pattern recognition molecules are present either as homodimers or heterodimers. For example, 442126, these are heterodimers, 55 homodimer, and 1111 is also homodimer. So these pattern recognition receptors, which are present over the plasma membrane of some immune cells or the host cells, they will recognize different kind of PAMPs, pathogen associated molecular patterns. Suppose the homodimer of TLR4, toll-like receptor 44, they will recognize viral proteins. If there is a heterodimer 2,1, a toll-like receptor 2 and 1, they will recognize bacteria. Oh, sorry. Actually, it is given in two other formats. One kind of receptors are present over the surface. Other type of receptor are present in the form of endosomal or lysosomal compartment, right? So the surface receptors or the surface pattern recognition receptors or toll-like receptors, they will recognize 2,1 combination will recognize bacteria and parasite. 2,6 it will recognize gram-positive bacteria and fungus. 5 is recognizing flagellin protein, so it will bind to the flagella of bacteria. 11 will bind with uropathogenic bacteria. There are few intracellular receptors, toll-like receptors. For example, this one, TLR33, which will bind with viral double-stranded RNA. 7 it will bind with viral single-stranded RNA. Actually, 7 and 8 both can bind with a single-stranded RNA. And there is TLR9. This is very important. It will recognize a bacterial and viral DNA, which is actually having CpG islands. So what are CpG islands? These are dinucleotide repeats in the DNA. This is CpG island. CpG means it is having C followed by a phosphodiester bond and then G. And if the DNA is having these structures, CpG islands, they will be recognized by a special class of pattern recognition receptors known as toll-like receptor, and the homodimer will bind to the CpG islands. So I think we should, there are 12 or 13 toll-like receptors. Okay, so there is a query also. What is the full form of CpG islands? So there's no full form. Okay, so CpG is a nucleotide. Okay, C is a nucleotide and C is connected to G. So let me illustrate this like this. We do have 4 bases in our DNA, A, T, G, and C, right? So these are 4 bases which are present, nitrogenous bases which are present in our DNA. Suppose we are having repeats of C and G followed by one phosphodiester bond. They are called CpG islands. Okay, they are characteristic of few pathogens. They are present in the DNA of few pathogens which are recognized with the help of this TLR9. So whether it is bacterial, so please note here, whether it is bacterial CpG DNA, it will also recognized by TLR9, or whether it is viral CpG DNA, it will also recognized by TLR9. So TLR or toll-like receptor 9 is responsible for recognizing these sequences which are present in the pathogen DNA. So So some few pathogens which are having these CpG islands or repeats in their DNA, they will be recognized with the help of this toll-like receptor, including both bacteria and viruses. Right. So they will be recognized by one class known as toll-like receptor 9, TLR9. I hope you are able to understand. So I will discuss them in detail in next class. I'm not going into the detail, but at least I hope you are able to understand the two terms. One is known as PAMP, okay, pathogen associated molecular patterns, which are present over, and these are present over microorganism, right? And there are pattern recognition receptors which are present over some immune cell which will bind to this PAMP, right? And these pattern recognition receptors are, one example is toll-like receptors, TLRs. And TLRs are of various types. So there are 1 to 13 TLR. Okay, we will discuss them in detail, and you need to remember, I will also tell a few tricks to remember them. Okay. For example, one you know now, TLR9 will recognize the CpG islands in the DNA. As I told you, the TLR7 and 8, they will bind with the single-stranded RNA only, right? TLR5, also we have discussed, they will recognize the flagellin protein. So because we know flagellin protein is present in the flagella, and flagella is a bacterial component, so they will bind to the bacteria, bacterial flagella, TLR5. Then we have TLR2. TLR2 is important because it will recognize the cell wall of bacteria, that is peptidoglycan. So it will recognize the peptidoglycan bacterial cell wall, right? And TLR4 is also very important because it will recognize the gram-negative lipopolysaccharides. So if you see this, it is written here. I cannot move the... Okay, so it is given here, TLR4, which will recognize the lipopolysaccharides. And you know lipopolysaccharides are present in gram-negative bacteria. TLR4, they recognize. So these are pattern recognition receptors. They are present over macrophages or some other immune cells of innate immune system, and they will recognize lipopolysaccharide, for example. These lipopolysaccharides are present in gram-negative bacteria, and therefore they are PAMPs, pathogen-associated molecular patterns. So if you are able to understand these two terms in today's class, pattern recognition receptor and PAMP, pathogen-associated recognition molecules or patterns, pathogen-associated molecular patterns, then in next class, I will let you know how they are able to recognize different kind of molecules. All right. You need not remember all of them, but some are very important, and therefore we will discuss important ones. Actually, there are 13 different type of TLR members. So 10, they are present in human from TLR 1 to 10, and 12 from 1 to 9, and 11 to 13. Actually, TLR 10 is absent in mice. So in mice, there are 12 toll-like receptors which are present. So all the toll-like receptors are actually the pattern recognition receptors, PRRs, which will recognize specific patterns which are associated with pathogen-associated molecular patterns, and therefore they are specifically able to bind the pathogen. जब क्लास स्टार्ट हुई थी तो मैंने बताया था they are not very specific innate immune cells are not very specific. They will bind, they will show same kind of response to almost all pathogens. But yes, they are little specific because they will recognize specific pathogen associated molecular patterns with the help of these pattern recognition receptors which are present over these immune cells. All right. If you have any query, please ask me. We will cover this again in the next class and then we will move. Okay. So there is a table in your QA also. If you have this reference book, you can go to page number 155 so that you are able to get this table. TLR1, for example, it is responsible for recognizing mycobacteria, gram-negative bacteria, right? And the component which is recognized is triacyl lipopeptides. Then second, I told you it will recognize gram-positive bacteria because it will bind to the peptidoglycan cell wall. So gram-positive bacteria. Not only this, TLR2 also recognizes zymosin, which is present in fungus. So gram-positive bacteria, fungus, yeast, all these are recognized by TLR2. TLR3, it will recognize double-stranded RNA. TLR7 and 8, they will recognize single-stranded RNA in viruses, mostly the double-stranded RNA and single-stranded RNA of viruses. So double-stranded RNA, 7 and 8, stranded RNA. TLR4, they will recognize the lipopolysaccharide, which is present in gram-negative bacteria. Not only this, they will recognize mannans and F proteins, which are present in some virus or in mannans, for example, in fungi. TLR5, very important. They will recognize the flagellin protein in flagella. Therefore, they will bind with any bacteria which is having flagella. TLR6, not very important though. Okay. 9 is very, very important. Asked several times because it will recognize the bacterial as well as viral DNA, which is having CpG repeats, unmethylated dinucleotides. And it can recognize herpes simplex virus components. It can also recognize hemozoin. Hemozoin is a digestion product of, or the byproduct of malarial parasite, P. falciparum, hemozoin. Actually, we also have some other TLR 10, 11, 12, 13. We do not know exactly where, what is the PAMP they are recognizing. We do know the pattern recognition receptors, but we do not know the PAMPs, pathogen-associated molecular patterns. But either 13, for example, they bind to the vesicular stomatitis virus or some bacteria, toxoplasma, or in some cases, we do not know in what kind of microorganism they recognize these patterns, right? So only 9 are very well studied and we know much about 9 TLRs, but exactly there are 13 toll-like receptors, 10 in humans from 1 to 10 and 12 in mice. Okay, so mouse is actually having 12 number of toll-like receptors recorded. All the toll-like receptors are pattern recognition receptors, as I told you, and they are having leucine-rich repeats, LRR. So it is also shown in next diagram as it is horseshoe-shaped extracellular ligand binding domain, right? So this horseshoe-shaped domain is shown here. If you have seen the horseshoe, this is leucine-rich repeat, which is present extracellular surface of any immune cell, which is actually having this toll-like receptors. So these are known as leucine-rich repeats or LRRs, which are extracellular recognition molecules. So these binding molecules will recognize, for example, TLR3. We have discussed the TLR3 will bind to double-stranded RNA, right? TLR7 and 8, they will bind to single-stranded RNA in viruses. TLR3 will make a homodimer. And then this horseshoe molecule, which is having leucine-rich repeats, and they will bind with this double-stranded DNA as shown in this particular diagram, this one. Right, so the TLR3 will make a homodimer. It will recognize double-stranded DNA in viruses, and this structure, which is a leucine-rich repeats, will bind to the DNA because They are very specific. They will bind to double-stranded DNA only. If there is a heterodimer, for example, TLR2 and 1, they will recognize lipopeptide. And this lipopeptide is bacterial triacyl lipopeptide, as it is shown in the, it is a combination of TLR2 and TLR1, which is given in your book also. So you can note it here. It is present here, TLR2 and TLR1. They will recognize bacterial triacyl lipopeptides. Lipopeptide means the combination of lipids and peptides. If you do not remember lipopeptides, these lipopeptides are present in gram-positive bacteria, as I showed you in previous diagram, right? So lipopeptides, sorry. Okay, lipopolysaccharides, glycoproteins, glycolipids. So, okay, you need to remember that these pattern recognition receptors will recognize one of them. These can be glycolipids, glycoproteins, lipoproteins, which is a combination of lipids and proteins, and so on. So lipoproteins are a combination of when the lipids are having bound proteins. And it is recognized by a heterodimer, which is a combination of TLR1 and TLR2. All right. So if you have query, please ask me. That's all for today. We will meet in our next class, and then we will discuss these receptors in more detail, and then we will move further.